Hello? Hello?
Online and also I can't hear you now.
Okay.
I'll try to speak louder.
Okay.
Very nice. Thank you.
Thank you for coming. I should stand there. Okay.
Good evening, dear students, colleagues, and audience online. Welcome to Professors of XJTLU Public Lecture. 欢迎各位莅临在西普听讲座。I'm Professor Beibei Tang from the School of Humanities and Social Sciences. It's my great honor to be the academic chair of this lecture series. Professors of XJTLU Public Lecture is our university's signature event for scholarly communication and knowledge dissemination. At XJTLU, now we have around 45 professors through whom we would like to promote our unique research culture. This public lecture series will be delivered by XJTLU professors from different research fields, introducing their career-long research or a comprehensive review of selected research topics, as well as the cutting-edge developments in the field. This lecture series will be offered throughout the academic year to both XJTLU staff, students, as well as the public audience. Through stimulating presentations using plain language, the professors of XJTLU Public Lecture Series will explain complex research topics and bring showcase of our university's research impact and its contribution to our community and society. Today's speaker is Professor Uromi Goodale from our School of Science. Professor Goodale received her PhD in 2009 from Yale University. From 2009 to 2012, she conducted her postdoctoral research at University of California, San Diego. In 2012, Professor Goodale made her journey to China. She was a postdoctoral fellow with the Chinese Academy of Sciences from 2012 to 2014, working on Xishuangbanna Tropical Botanic Garden. From 2014 to 2022, she worked at the Guangxi University in China. In 2022, she joined XJTLU's Department of Health and Environmental Sciences as a professor of plant ecology. Professor Goodale's research focuses on seed biophysiology, ecology, and conservation. She is the editor-in-chief of the Journal of Sustainable Forestry and the chair of the ecology section of the Botanical Society of America. In today's lecture, Professor Goodale will lead us to explore the mysterious world of orchids and discuss some of the secrets of learning how to contribute to their conservation. Professor Goodale will speak for about 15 minutes, and then we will open the floor for questions and discussions. Without further ado, I give you Professor Goodale.
Hello, everyone. Good evening. Can you hear me? Wonderful. So thank you for being here. I'm delighted to be here today. But before I begin, please allow me to thank the leadership of the Xi'an Jiaotong-Liverpool University for inviting me to this distinguished lecture series as a speaker. And I would also like to thank Professor Bei Tang for that wonderful and kind introduction. In this talk, I will delve into the biology of orchids so that we get familiarized with their flowers, roots, plants, seeds, and then also introduce some of the research that was done in my lab that focuses on regeneration ecology, seed biophysiology, and conservation, and maybe also learn a little bit about how we, as individuals, as a society, can contribute to the conservation of this fascinating plant family. Let me see how I skipped. Okay, wonderful. So our experience in what orchids are like is usually something like this. Orchids that are commercially bought are meant for display. They have this long stalk stem clipped together. So we have this beautiful flower standing up straight that can be on top of our beautiful table. But in nature, it's not like that. These stems fall off, and then they hold their flowers in front of the plant, allowing the pollinators to come close to them. Orchids are quite close to my heart, and I have a lifelong passion with them. From childhood, I was very interested in these plants. And around age 10, growing up in this tropical island, Sri Lanka, where orchids grow amazingly well, I began growing orchids. By about age 12, I was able to figure out how to grow them, having them on a wooden mat with a green shade cloth, about 1.5 meters high above ground. And I used charcoal. Can someone help me with this? It's not moving. Hold on a moment, we will figure out how to move the slides forward. Thank you. Yes, so let's see if it's going forward. Wonderful. So this was my, the very first orchid that I grew. These purple, beautiful dendrodiums that started in very small, tiny charcoal pots. And I consecutively moved them into bigger pots until they became to flower. And this gave me much joy. It was a triumphant moment in my life as a budding plant ecologist. At that time, I had no idea that I would spend the rest of my time, my career as a plant biologist focusing on plants. So beautiful, huh? Showy, fascinating, attractive, bright. Let's talk about this flower. This is the thing that makes us go for orchids. Some people spend much of their money collecting these rare orchid species. This flower has 3 sepals, 3 petals that include the labellum and the central column. And this central column has the anther and the pollinia and the stigma. In terms of the variation that you see in flowers, these are the main components in terms of size, color, shape, texture, as well as scent that give the diversity, the very high diversity to the orchid family. So I press this one, nothing happens. So I should use that? Okay. So the diversity that you see in these flowers is linked. The diversity that you see in these flowers is linked to specialized pollination mechanism. That's number 1. The second one is symbiotic associations with mycorrhizae. The third one is the colonization of epiphytic plants. And the fourth is a special kind of mechanism used for photosynthesis called cressulation acid metabolism. We'll go through this little by little, one by one. When it comes to scent, you can see this in this graph that 2 flowering orchids of the same genus, the Gymnadinia species, has 2 signatures for the compounds that give the scent. So this can lead to having different pollinators that are sensitive, that are able to be attracted to these scents to come towards them. The second one is the associate pollen delivery that is mediated by having special pollinia that appear in different places. You can see from these arrows, again, same genus, Platanthera species, but the pollinia are in different places that can attract different pollinators that are matched for the size. So the sticky viscida, shown in yellow arrows, this placement allows the flowers to differentiate. And when it comes to the third mechanism, which is where how the flower morphology changes, the morphological isolation of 2 species in the Gymnadia with different spur lengths in their flower shows that species that have a longer fitting tongue could easily access the nectar here, whereas the species with the shorter fitting tongue can attract the pollinator here. That again gives diversity to the visitation of pollinators with different tongue lengths. Thus, different pollination syndromes have given rise to a variety of orchid species. Orchidaceae appears to have undergone divergence in 2 places with significant acceleration of net species diversification in the Orchioides and 2 accelerations in the upper epidendroids. I'm not going to go into a lot of detail here, but I want you to see these next few slides from my PhD student that are images of some of the most amazing orchid flowers that I have seen. Today, this family contains more than 750 described genera and more than 28,000 species. In addition, there are 200,000 currently identified hybrids, making it a hyperdiverse family. The demand for orchids in quantity and variety has increased year by year, forcing scientists and breeders to develop new varieties using crossbreeding, selection breeding, mutation breeding, molecular marker-assisted breeding, transgenic breeding, and genome editing breeding. Flower morphology or color, scent, flowering time, abiotic stress resistance, these are the most important characteristics that are targeted in breeding systems of orchids. However, the identification of gene function is a key prerequisite, although currently there's no stable transformation system available for orchids. Current studies mainly achieve transformation using agrobacterium-mediated procedures and particle bombardment and gene slicing. Also, transcriptome data is very limited. So this is an important area of research that we can go forward in developing new varieties of orchids. Results of crossbreeding in two species of orchids that have very different flower color, the purple here and the yellow here. You can see the final result is Alomasia popcorn haruri, and its parents are here. But you can see that the crossbred flowering has subtle characteristics of both plants, and this is an area of research that's future is very open. They are beautiful and subtle. So this is one important way we can bring forward orchid conservation. Orchids do not have diversity in these flowers. They're also very diverse in their habits. Some are terrestrial species that are grown on the ground. Endangered terrestrial orchid, Plateaula sterata, you can see almost like a planted garden, but this is naturally growing in Yaqiang Natural Nature Reserve for orchid conservation in Guangxi, China. One of the two nature reserves in the world that is dedicated specifically for orchid conservation. Habitat diversity is also represented by epiphytic orchids and lithophytic orchids. Epiphytes are growing on top of tree trunks, such as what you see in this image. And lithophytic orchids can grow on rocks, sometimes covering the entire rock. You cannot see it here, but this is an entire rock covered by a Paphiopedilum hirsutisum species. 70% of the orchids in the Orchidaceae family are epiphytic, compared to only about 10% in the entire flowering plant families. Others can grow underground. They cannot be seen until it is flowering time. They are called saprophytic orchids. They grow on soils which lack nitrogenous nutrient compounds, so they fulfill their nutrition requirement with the help of mycorrhizal fungi. Their leaves are reduced to mere scales, or sometimes they are leafless, and they do not process true roots. Instead, they have rhizomatous or fleshy fibrous roots. This is a spotted corolla root orchid. It is a saprophytic orchid with small brownish flowers with pink spotted white lip. The orchid garden I mentioned before, the Yaqiang Reserve, is an ideal site to conduct research on orchids. It's located in Guangxi province in Lehigh County. Although the thick humus soil is retained where the terrain is depressed under the forest, generally this terrain is very rocky. It's difficult to cultivate these species, duplicating these natural conditions. And conservation is much preferred in situ. So safeguarding these places that are really important for orchids is a very important thing. In Jiaotong, although orchid species are concentrated in the small area, their population size are not even. In general, the population of epiphytic orchids tend to be higher in abundance, much high in numbers and density, so thick carpets, while the terrestrial orchids tend to be much fewer. So this is an important question that my PhD student tried to ask. That's what she studied, Young Mei. She was the former deputy director of research for the Jiaotong Orchid National Nature Reserve. She studied the ecophysiological significance of photosynthetic nitrogen use efficiency and nitrogen allocation. That means when energy is harvested, how nutrients are connected to that process in an orchid community in a karst limestone region. She focused on 28 wild orchid species, 6 of which were terrestrials and 22 were epiphytes. She explored their ecophysiological characteristics in order to see what the different strategies are that are adopted by sympatric, that means growing together, orchid species that are terrestrial and epiphytic. The objective was to understand the functional factors that may facilitate this dense distribution of epiphytic orchid species in this kind of habitat. So what did she find? She conducted both field and greenhouse experiments. After the field experiments were done, some plants are moved to the greenhouses in Nanning Botanical Garden. She measured things such as their maximum photosynthetic rate. And after the measurements, she took leaf samples, then collected and tested these in the lab to get other characteristics such as leaf area, leaf mass, chlorophyll content, and she tried to figure out what the nitrogen use efficiency was in these species. Now, let's look at a few of her results. Her results showed that the maximum photosynthetic rate of these 28 orchid species were low compared to other plant species. So orchids kind of slowly doing their thing. They're limited by light, they're limited by nutrients. But what she did find out was that the maximum photosynthetic rate, photosynthetic nitrogen use and leaf nitrogen content of the terrestrial orchid species was higher than that of epiphytic orchid species, agreeing with the hypothesis that epiphytic species would have greater functional capacity compared to the terrestrial species that live on the forest floor. So here's the photosynthesis, nitrogen use efficiency, nitrogen content, and epiphytes are here and terrestrials are here. The fraction, the amount of leaf nitrogen was allocated for the photosynthetic apparatus, that did not show any difference between epiphytes and terrestrials. But the nitrogen content that you see in the middle graph here in the photosynthetic apparatus of the epiphytics were lower than that of terrestrials. Furthermore, when you consider the whole leaf, the leaf mass per area of epiphytic orchids was significantly higher than the terrestrial species. What does all this mean, these characteristics? This basically means that epiphytic orchids adopted a survival strategy of low growth rate, strong stress resistance, allowing them to be better adapted to the harsh, karst, rocky habitat. So they can survive and flourish in this kind of habitat. One other important mechanism is that they have driven the diversification of orchids is the crassulation acid metabolism. It's a kind of photosynthesis. We call it CAM photosynthesis. It's a method of carbon fixation evolved by some plants in dry circumstances. In most plants, the stomata, that means the small tiny pores on their leaves, are like mouths that take in oxygen all along the surface of these leaves. They open during the day to take in carbon dioxide and release oxygen. plants must take carbon dioxide because they use it as a source of carbon and build sugars, proteins, nucleotides, basically the molecules that are needed for building the plant body. They must also release waste oxygen, which is the byproduct that is left over from photosynthesis. Most plants open their stomata during the day because that is when energy is received from the sun. The energy from the sun is harvested by the chloroplast and used to make ATP, a compound, and NADPH, another compound. And these short-term energy storage molecules are then used to power the fixation of carbon into sugar. In plants living in very dry environments, what does this mean? You keep your plant doors, windows open. So it tends to be a way that you could easily get dried up. So what they do in order to meet their needs to continue the sun's energy with carbon dioxide from the air, CAM plants, the crassulation acid metabolism photosynthesizing plants, take their carbon dioxide at the time when we are all sleeping, at night, and store them in the form of a 4-carbon atom that is called malate. The malate is then released during the daytime, slowly, so that it can be used for actual photosynthesis, for ATP and NADPH creation from sun's energy. Now, this allows a wonderful way for plants to be cool during the daytime in hot, dry conditions. The name this crassulation acid metabolism comes from the crassula plant in which it was first discovered. Many orchids can use this mechanism for photosynthesizing, and they can use the whole plant. This is what's amazing about orchids, not just the leaves. They can conduct this photosynthesis in their whole plant body, in the roots, in the stems, in the fronds, and sometimes even in their flowers. So orchids are really amazing. Not many places have completely assessed how much percentage of the orchid plants go into crassulation acid metabolism, but in Panama, it was estimated that about 10% of the orchids in Panama has this CAM photosynthesis mechanism. Orchids, especially the epiphytic orchids, have aerial roots. These roots hold fascinating biological relationships with mycorrhizal fungi. They are also the site for another source of orchid diversity. The fungi can be symbionts that develop mutually beneficial relationships with the plants. These relationships are ubiquitous, found everywhere in nature. They develop key interactions in building ecological communities. Let's look at the root a little bit more. Mycorrhiza associated with plant roots affect plant and population characteristics. They provide resistance to pathogens and herbivores, affect distribution of non-native and native species, and allow growth on malnourished substrates. In orchids, the endomycorrhiza, endo in Latin means inside, that live inside the plant cells within roots can improve the disease resistance of host plants and assist the plants to absorb nutrients. They're coiled up inside cells here. In this next figure, you can see that a little bit more clearly. Due to the important ecological and evolutionary significance of this association, much research has been conducted on this relationship. But we don't know everything. And one of the interesting things is when sympatric species that are grow together, do we have the same fungal association in these two plants that are very related but growing in the same conditions? This was the research question that was asked by one of my students. Many adult orchids, especially photoautotrophic species, associate with a diverse range of mycorrhizal fungi. But we don't know whether these associations change with time, whether they actually are different in different species, and functioning of orchid mycorrhiza during vegetative and reproductive time, whether that is different. So Wang Xiaoying, another PhD student of mine working with me, focused on the seasonal dynamics of endomycorrhizal fungi in two congeneric orchid species that are grown also in this Jiaotong National Natural Reserve. You can see here some of the isolated fungi that she was able to build in her research work. She focused on two species, Paphiopedilum dianthum and Paphiopedilum hirsutisum, and she explored their seasonal abundance and diversity, focusing on fungal species by trying to extract them and identify them, as well as do complete plant fungal analysis using genetics. She combined next generation sequencing methods and isolation of fungi. The collected root tissue samples were first surface sterilized by her to scrape out the pelletons inside the cortical cells to check if there is pelleton colonization, and if they were there. Then she extracted the pelletons using specific tubes directly from the roots under microscope and deionized water to clean them 3 times. Then they were placed on specific fungal culture media to grow for about 1 week. After they were growing for such a long time, such a short time, about 1 week, they were subcultured to fresh media, and after about 3 to 4 weeks, they were subcultured again until she was able to isolate them. And finally, she transferred them into liquid media to prepare for DNA extraction. Extracting both from this method as well as the direct root was sent for high throughput sequencing, and the isolated samples were sent for single strain identification. So we had 2 sets of data that came out of this work. What does that show? Her results show that fungal community of both species were dominated by the phylum Ascomycota. About 93% belong to this phylum. The most abundant fungi present in all four seasons in terms of time belong to the Sodariomycetes class. What about the 2 species? Paphiopedilum dianthum had higher richness and diversity of endomycorrhiza compared to Paphiopedilum hirsutisum. These are 2 diversity indices, and these are the 2 species that you find here. But there was a difference in the seasonal response in the 2 species. Paphiopedilum hirsutisum was sensitive to seasonal changes, but dianthum was not. These studies require highly trained scientists. It takes me a few slides to show you these results, but it's a lot of very detailed work. Just pulling out the peloton from a very thin tube going into a single cell required her to get trained, and we had a scientist from Florida International University who came over to support her research. It not only gives results to understand the processes, but these isolates that we develop from these kinds of studies are a very important resource for medicines and other users. A huge resource waiting to be discovered in this hyperdiverse Orchidaceae family, and an opening for research and studies. So we looked at orchid flowers and the growth habits and roots a little bit now. Let's look at another fascinating feature of orchids, one of my favorites, their seeds. Compared to other seeds, orchid seeds are very small. They're called dust seeds. They're like dust. They're like powder, but under the microscope, they reveal a beautiful structure, as you can see here in the Xi'an species seed. It is a saprophytic orchid. Compared to all the seeds that you may be familiar with up here, the scale here is 100 millimeters. The same scale here is 100 micrometers. So they're very, very small. This shows you the kind of diversity of seed size we see, and this is a log scale down here in the plant family. So in orchids, the seed size can vary from about 0.05 millimeters in length, and they have about 120 to about 180 cells. Not very big. There's not much that you can store in such a small seed. And there's huge variety in the shapes and sizes of these seeds. The epiphytic species are shown up here, and then we have the terrestrials in the middle and saprophytic species. You can see they can be like wormy, spindular, more globose, many, many beautiful kinds of shapes are here seen in this diversity of orchid seeds. What's amazing is that these seeds come inside an orchid pod. The pod has millions of seeds. Each one of these is a seed. It opens up by dehiscing on the sides, and when the conditions are right and the dryness is right, these seeds go into air, and these are the dust seeds of orchids. Not only is the seed very different in orchids, orchids are not like other plants when it comes to the germination of seeds. We are used to seeing seeds germinate. The first thing we see in other plant species seeds is a root coming out. But in orchids, roots come last. The biology of seed germination requires a fungal symbiont to facilitate the process. So during germination, the fungal hyphae make contact and enter via micropores into the minute seeds. They penetrate the germinating embryo and cross the plasma membrane of the cortical cells to form an intracellular, inside the cell, structure made of masses of fungal hyphae we call the peloton. The pelotons are subsequently digested, providing food for that growing plant. So the fact that orchid seed was a tiny small seed, didn't have much in terms of nutrients, doesn't really matter. This fungi is able to give that resource to the growing plant, increasing the seed's tolerance to environmental stresses and facilitating the establishment and colonization of photosynthetic plants, these plants, in new areas. So once the photosynthesizing happens in the orchids, it goes through the growth process and comes up with the plantlet. Sometimes the plant is able to give back resources to the fungi. This symbiosis is known to help seedlings to grow better and faster, establish well when transferred to soil, and to be better controlled against future pathogens compared to seeds germinated without fungi. However, these benefits depend on the encounter probability with suitable fungi. Because of this reason, orchid seed germination is different, and they have partners that help them. So they don't develop roots first. It's fascinating difference when it comes to orchid plants. The roots come later. Seed conservation is an important ex situ method for saving threatened species. A tiny seed, one seed can give a small, tiny plantlet. And this is what my third PhD student, she has graduated now, Dr. Namrata Pradhan, focused on. She investigated the orchid seed biophysiology for conservation through long-term storage using cryopreservation. She focused on 20 orchid species and investigated 5 of them were epiphytic and 15 was terrestrial. She investigated the suitability of viability tests, which means a test that allows us to understand whether this orchid seed is live or not, and methods for seed conservation, and develop a comprehensive methodological guide for seed conservation. So these viability tests, you first do the sterilization, and then you do some washing, and after that you use the tests. She looked at three different tests: TTC, FDA, and Evans Blue test, and then finally evaluated under a stereo microscope, taking a photo and digitizing that photo to see which tests were best suited for what kind of species. Seed viability testing of orchids is understudied. That's one thing that she found through her literature review, which prevents efficient conservation of threatened orchid species. The tetrasolium test is the most commonly used seed viability test, but it was most unreliable in most studies. The FDA or fluorescent diacetate test and Evans Blue test are claimed to be more reliable than the TTC test, but her literature review showed that they were used in very few studies. The studies with orchid seed viability tests were not always accompanied by a germination test that can be compared when it was done. And sometimes you would find very conflicting results when you compare with the chemical test and the germination tests. Here you can see that there are these three tests that were used on epiphytic orchid species and the same on terrestrial orchid species. You can see without sterilization and sterilization, there's a slight difference, and that when it comes to the three different tests, the results were different. The Evans Blue test was the best in both kinds of orchids, and these were results that were first for the orchid community, 20 species, and her study has inspired other researchers to use this standardized methodology to conduct and compare among a large group of orchid species. The number of orchid species investigated for seed viability is considerably low, despite the fact that orchids have one of the largest flowering plant families, 28,000 species. So the Evans Blue test was generally much better, as we saw in the results, but also the type, that in addition to the test type, life form of the species and the seed sterilization were influential factors that determined seed viability testing. Namrata compiled a large amount of literature from knowledge, using also her knowledge that she gained firsthand by working in this species in the field and in the laboratory during the course of her PhD. The seed conservation protocols she established in the studies of this thesis were also used to give a comprehensive idea of how do we conserve seeds. There were also communication with experts in the field of orchid conservation, and finally she put all this data together to combine and develop a step-by-step comprehensive methodological guide that can be used by conservation biologists. And this guide has 6 successive stages of orchid conservation that were identified, each stage with many different phases. Stage 1 was basically focusing on field work, and it's important that before we begin any seed conservation process or program, to take the necessary proper permissions to do the collections, that we follow all legal formalities. And if we are going into native lands, also get permission and follow societal guidelines. It's important to familiarize oneself with the legal rules. The second step is prospecting for orchid populations in the field, usually to locate the target species and record the data for that species. We usually take a local naturalist or other expert with us who has experience as a field guide. And the third step is identification of the species. And the two main methodologies are morphological and molecular identification. The morphological identification involves preparing herbarium specimens, identifying the flora in the field using guidebooks, color photographs of herbarium sheets as references. If this is not successful, taxonomist or a specialist in the local flora can also help. For molecular identification, the DNA barcodes are used. So we get permissions, we follow the process, and then we go towards the third step to do the identification. When this is done in stage 1, we move towards completing the field work and move forward towards completing the field work. When we finish this, we go towards deciding for species selection. So it's important to understand what species are we working? Is it threatened or not? Are there enough individual plants for sampling? Has it been already sampled in other places? And asking these questions make us be more aware of what we take out of the seed. And when it comes to the seeds, in the specific species that we've identified or targeted for conservation, this is also an opportunity to gather as much data of the plants as possible in the field. And then we need to conduct a seed pod assessment, because not all seeds are inside the pod are ready. So when is the pod holding these millions of seeds, when is it ready? Just before dehiscence. And some of these pods mature across years, sometimes 2 years. So really understanding the species is really important to collect the seeds at the right time and bring them back for conservation. We usually check the conservation status, and also the CITES status, and we have to have the right permissions to get the seeds collected. All these phases sum up stage 1. So Namrata departed this process into 6 stages. Basically, stage 1 is something that happens in the field. It is field work. After the species is identified and we consider all these factors, here we are summarizing it again. We have a good idea. We have completed our field work. We move on towards the other stages. The other stages, stage 2, 3, 4, and 5, they are mostly work that is done experimentally assessing the orchid seeds in the laboratory after they have been collected. So they are brought into the laboratory as mature seed pods, and we open them under sterile conditions as soon as they are brought in. The seeds are extracted under a laminar flow cabinet, and they're placed in sterile vials. Usually the moisture content of the seeds are checked before they are typed for research or seed conservation. For most orchid seeds, it's crucial to dry them so that they can withstand storage, so that they can remain viable. And how much to dry them? Each species may have very specific requirements, so it requires us to take a bit of work to do this. Earlier we saw that there are specific tests to use, so which test is the best for viability testing we need to know. And when we go towards the latter stages, sometimes we may need to consider the symbiotic germination, because the orchid seeds may not be, it may not be viable to germinate them without the fungi. So it's a very engaged, involved process of different stages. We may have to test media in order to know, which ones can germinate in which kind of media. And orchid seeds can be basically stored in 3 types of conditions. Sometimes it's just a matter of putting them in the fridge. Sometimes we may need a freezer. And other times, we may use very low temperatures with liquid nitrogen, such as minus 196 centigrades, which we call cryopreservation. So storage temperature is an important factor that influence the seed viability and germinability, and in turn, how long, the longevity of storage. Orchid species have shown that there is variation in longevity when stored at different temperatures. Therefore, each species may need to be individually tested for the right conditions. Now you can see 28,000 species, not more than about 160 species do we know really clearly all of the process for this that we are talking about here. So it's still an important research area that is open in the orchids family. So here I summarize stage 2 again, basically going through 2, 3, 4, 5, and 6th stage, which is finally seed banking until you are ready to take them out for research or for generating new plant species, new plants for conservation. So the latter stages are basically laboratory work. 2 to 6 focuses on the laboratory work. Despite the importance of plant diversity for human well-being and the threats for plant survival, plant conservation gets much less attention and support compared with vertebrate conservation. That is a reality for us. Here you can see a beautiful picture of plants that are conserved in situ in Xishuangbanna Tropical Botanical Garden, where I did my first postdoctoral research, my first arrival, my first home in China. Plants can be conceived, should be, where possible. That should be our first priority, to conserve them in situ. Here is the Jiaotong Orchid Nature Reserve that shows about 40 different orchid species are here in this picture. They can be in protected areas and also sometimes in living collections, seed banks, cryogenic storage. At least one option is available for all species for conserving them that need it. Maybe no single method works for all, so we need to figure out what is the best situation. The major bottleneck, and that's where my work, my career comes into play, is the shortage of skilled people. New technologies, machine learning, and citizen scientists can extend the research of experts. These are summarized ideas that were presented by Professor Richard Corlett in his recent analysis that put together what we should do to prevent extinction, zero extinction. So training initiatives, young people, you in this audience, we are the future, so that we can increase the probability of saving these plants, decrease the zero extinction. When it comes to orchids, orchids face even more significant biotic losses due to illegal extraction, overharvesting, habitat loss, and detrimental effects on populations from climate change. Here, in this next picture, you can see, despite orchids being the flagship species in conservation ecology, while despite having lots of rare species, it is most affected by climate change. Because, as I told you, the pollinators, it is very specific. The fungi can be very specific. And these very intricate relationships with other taxa in orchids, everything has to be kept in a real good balance in order for everything to work well. And now, Orchidaceae is one of the most affected plant families. Here you can see some wild orchids that are sold in medicinal markets. They are directly extracted and brought to the market. So in China, many of my colleagues who work in orchid conservation in many of the botanical gardens have really done an amazing amount of work to bring methodologies that can grow these orchids for medicinal purposes in large scale But we still have to overcome the idea that such grown plants may not be as medicinally valuable. So sometimes we do research and a lot of education to let the people know that this is something we should do. The whole family of Orchidaceae is listed under Appendix 2 of the Convention of International Trade of Endangered Species, CITES, of wild fauna and flora. In China, many formerly common orchids are now listed locally endangered in the Chinese Red List. In situ conservation of threatened taxa, such as orchids, takes priority in the management. So habitat conservation is very important, and to prevent species extinction. And that is why one of my favorite places in China, Jiaotong Orchid National Nature Reserve, is a very, very important place. So I invite you to explore for your next visit this amazing place. It's dedicated, as I said, for conservation of orchids. There are 438 tropical and subtropical orchid species and high abundance of plants, out of which in China, out of which Jiaotong has 147 species. In situ conservation may not be sufficient, so we have to come up with a very comprehensive approach when it comes to deciding on orchid conservation. That is why I will leave you with these last two slides, that individual responsibility, so that we reduce the pressure on natural resources, on land. Adhering to very sustainable, good resource consumption practices are very important. I tell my students, we talk about the 6 R's: reduce, reuse, recycle, refuse. If you don't need it, do I really need this? Don't take it. Rethink before you throw something away. Repair, get the skills. These are the critical thinking skills, but they need to be critical life skills that we make second nature in our daily life. So that's one way we can contribute. Getting involved, learning more about our natural resources is important. I hope that I was able to convince you through these beautiful photos taken by my PhD students of this fascinating plant family that we only have one planet to live in. And in it, we are interconnected. We are part of it. There's no plan B. It's our responsibility to contribute to the sustainable world and continue to be able to enjoy this fascinating plant family. Thank you very much for listening. I thank my students, my collaborators, XJTLU, funding organizations, for this amazing opportunity given to me as a research scientist to focus on plants, one of which, a major part of which is orchids that I try to present to you today. Thank you very much for being here. Thank you.
Thank you, Professor Goodale, for your fascinating talk. Thank you for showing us the mysterious world of orchids. Dear audience online, we will stop our live streaming here. And thank you for staying with us. And we look forward to seeing you next time. Thank you. And for the audience in the room, we will open the floor for questions and discussions now. So if you have any questions, please signal.
I'm sorry about the technical difficulties at the beginning, but I hope that I was still able to inspire you. So any questions from the audience?